In 2023, an AI designed a brand new antibiotic from scratch in 48 hours. Not a variation of an existing drug, not a tweak to something already approved. A genuinely novel molecule, targeting a bacteria that had become resistant to nearly every treatment medicine had produced in the last 50 years. The researchers tested it. It worked. That compound, which no human chemist had ever imagined, came from a model that had never once sat in a lab, never pipetted a solution, never stared at a failing patient chart. It simply thought its way to an answer that a century of human effort had missed. Here's why that should stop you cold. Right now, approximately 10,000 diseases have been identified in humans. We have effective treatments for fewer than 500 of them. That gap, 95% of human disease sitting essentially unanswered, represents not a lack of effort, but a fundamental limitation in how the human brain handles complexity. Drug discovery, at its core, is a combinatorial problem. The number of possible drug-like molecules that could theoretically exist is estimated at 10 to the power of 60. That is not a large number. That is a number so incomprehensibly vast it makes the total atoms in the observable universe, 10 to the 80, look almost manageable by comparison. For decades, pharmaceutical companies have been exploring this molecular universe with the equivalent of a flashlight. AI just turned on the sun. The financial stakes alone are staggering. The average drug takes 12 to 15 years to develop and costs approximately $2.6 billion to bring to market. 90% of drug candidates that enter human trials fail anyway, meaning the industry spends billions on drugs that never help a single patient. The United States spends nearly $4.5 trillion per year on healthcare, a number climbing every year as diseases like Alzheimer's, antibiotic-resistant infections, and rare genetic disorders consume more resources with fewer solutions. Meanwhile, the global burden of disease, the years of healthy human life lost to illness, is measured in the billions annually. Alzheimer's alone affects 55 million people worldwide, and as of 2026, there is still no drug that stops it. Only 2 approved therapies slow it slightly in some patients. Treatments for pancreatic cancer, amyotrophic lateral sclerosis, and most parasitic diseases affecting the developing world have barely moved in decades. The cost of inaction is not abstract. It is measured in funerals. Understanding how we got here requires stepping back about a century. Traditional drug discovery works like an extraordinarily expensive and slow treasure hunt. Chemists synthesize a molecule, biologists test it against a disease target, usually a protein, and if it fails, they synthesize a slightly different molecule and test again. This process, called high-throughput screening, can evaluate millions of compounds per year, which sounds impressive until you realize that millions against 10 to the 60 is the equivalent of reading one page of a book that is longer than the known universe. The Human Genome Project, completed in 2003 after 13 years and $3 billion, gave researchers something crucial, a map of every gene in the human body. That map revealed tens of thousands of potential disease targets, proteins, enzymes, receptors that could theoretically be switched on or off to fight illness. The problem was that knowing a target exists is very different from knowing how to hit it. That gap between target and treatment is where drug development has always broken down. Proteins fold into complex three-dimensional shapes, and a drug only works if it fits that shape with extraordinary precision, like a key not just entering a lock, but triggering the exact right mechanism inside it. The first revelation came in 2020, when DeepMind's AlphaFold solved what scientists had called the protein folding problem, one of the grand challenges of modern biology. For 50 years, researchers knew that a protein's function is determined by its three-dimensional shape, but predicting that shape from a gene sequence alone had defeated some of the most powerful computers on Earth. AlphaFold predicted the structure of nearly every known protein with an accuracy that matched expensive, years-long laboratory methods. Within 2 years, it had mapped over 200 million proteins, essentially the entire known protein universe. To put that in scale, before AlphaFold, science had laboriously determined roughly 170,000 protein structures over several decades. AlphaFold did 100 times that number in months. Every one of those structures is a potential disease target. Every one is a door that was previously invisible. Researchers at the University of Toronto used AlphaFold data to identify a promising target for a rare pediatric cancer that had been effectively untreatable, and moved from target identification to a testable drug candidate in under 18 months, a process that would previously have taken a decade or more. The second revelation is stranger and more unsettling. AI is not just finding drug targets faster. It is inventing biology that does not exist in nature. A company called In Silico Medicine used a generative AI system to design a drug for idiopathic pulmonary fibrosis, a fatal lung disease with no cure, from scratch. From identifying the target to generating a clinical candidate took 18 months and cost approximately $2.6 million. The same process at a traditional pharmaceutical company typically takes 4 to 5 years and costs between $300 and $500 million. That is a reduction in both time and cost of roughly 95%. In Silico's drug, INS-018-055, entered Phase 2 clinical trials in 2023. But here is the part that genuinely reframes everything. The AI did not search a library of known compounds. It generated a molecule that had never existed before in any lab, in any living organism, at any point in evolutionary history. Evolution had 4 billion years to explore molecular space and never found this structure. The AI found it in weeks. This means the constraint was never nature's creativity. It was ours. The mechanism behind this is worth understanding properly. Modern drug design AI systems are trained on enormous datasets, millions of known molecular structures, their properties, their interactions with proteins, their toxicity profiles. The model learns a kind of deep grammar of chemistry, which arrangements of atoms are stable, which are toxic, which bind tightly to which protein shapes. Think of it like a musician who has listened to every piece of music ever composed, until they have internalized not just songs, but the underlying rules of harmony, rhythm, and tension. When you ask that musician to write something new, they are not randomly guessing notes. They are generating something coherent and novel within a vast learned structure. Drug design AI works the same way. Given a protein target, the system generates candidate molecules by navigating what researchers call chemical space, not randomly, but intelligently, guided by learned patterns of what tends to work. The counterintuitive part is that the AI does not understand chemistry the way a chemist does. It has no model of electrons or quantum mechanics in the way a scientist would articulate it, and yet it produces molecules that work. Understanding why remains an active and genuinely unresolved area of research. The complications are real and worth naming directly. First, AI-generated drug candidates still have to survive the same brutal gauntlet of clinical trials as any other drug. Generating a promising molecule is stage 1 of a very long race. Most will still fail in Phase 2 or 3 trials, and no AI system has yet cracked the harder problem of predicting how a drug will behave in an actual human body with all its variables. Second, the technology is concentrating power rapidly. Large pharmaceutical companies are acquiring AI drug startups at an accelerating pace, which risks gatekeeping these tools behind the same pricing structures that have made existing drugs unaffordable for millions. A 2025 analysis found that the 10 largest pharmaceutical acquisitions of AI companies totaled over $40 billion in 18 months. Third, regulatory bodies like the FDA have no established framework for evaluating drugs whose design logic cannot be fully explained even by their creators. Approving a drug you cannot entirely account for is a genuinely hard ethical and procedural problem with no clean solution yet. For you, the timeline on this is shorter than most people expect. If you work in pharmaceutical research, computational chemistry, or clinical trial management, your workflow is being restructured right now, not in 10 years. Companies including Recursion Pharmaceuticals, Exscientia, and Relay Therapeutics are already operating drug discovery pipelines that are majority AI-driven. The drugs in early trials today for Alzheimer's, antibiotic-resistant tuberculosis, and several rare childhood diseases include AI-designed compounds. By 2030, analysts at Morgan Stanley project that AI-assisted drug discovery will compress average development timelines from 12 years to under 5. That means drugs for conditions currently considered untreatable may reach patients within the decade. If you or someone you know is living with a disease that has no good answer today, that timeline is not an abstraction. Zoom out far enough, and what you are watching is a fundamental change in the nature of medical progress. For most of human history, we discovered drugs by accident. Penicillin found in a contaminated petri dish, aspirin derived from willow bark. Then we got systematic, but we were still limited by what human minds could hold and imagine. AI does not replace the scientist. It expands the aperture of